I wanted to welcome everybody to the Center on Addiction and Behavior Change seminar series, Spring 2021. We're well into our series now, and before I introduce today's speaker, I just want to highlight the upcoming, we have two more seminars in April. On April 14th, we're going to have our own Nicole Schramm-Cepeda from Duke talking about the relationship between mental health services and law enforcement in Durham County and substance abuse. So how we could change the paradigm in terms of treating illicit drug use to be more directed towards treatment rather than the drug law enforcement aspects of it and how they could work together. And then on the 28th, our last seminar is going to be also from a Duke person. Isaac Lipkis is going to talk to us about tobacco smoking among young adults and the perceived risks. Now, I am putting together the fall seminar schedule, and we're going to maintain the Zoom as far as I can tell from the policies of the university. And so one of the advantages that does have is we can have lots of outside speakers, and it's kind of a less expensive option in terms of having people sit at their desktop and provide us with their insights and research. And so if you want to recommend somebody, just let me know at ed11@duke.edu, or if you want to give a talk yourself, we still have some slots available. So now I'd like to introduce today's speaker, Dr. Kathleen Kantak, professor of psychological and brain sciences at Boston University. And Dr. Kantak earned her PhD at Syracuse University and is the founding director from 1982 of the Laboratory of Behavioral Neuroscience at Boston University. And she's quite active in undergraduate programs in neuroscience and especially relevant to our group at the CABC is involved with the transformational training program in addiction science at Boston University. So training the next generation of researchers and clinicians. So she's going to speak to us today about her latest research on extinction memory enhancement for cocaine relapse prevention, pharmacological, behavioral, and molecular determinants using an animal model of cue exposure therapy. Kathleen.
Thank you. Okay, I wanted to thank Ed for inviting me here today to share with you my work on extinction memory enhancement for cocaine relapse prevention. This project has been ongoing since 2007, and these are the various individuals who have contributed over the years. This work was supported by grants from NIDA, the NSF, and the Center for Neuroscience at Boston University. In animal models of drug self-administration, the addiction cycle consists of acquisition and maintenance phases, followed by extinction training and reinstatement testing to study relapse behavior. The emphasis of efforts for medication development and treatment has been directed toward the maintenance and reinstatement phases, with little directed toward the extinction phase. In 2002, Conklin and Tiffany applied extinction research and theory to cue exposure addiction treatments. They reported that although CET works well as a standalone treatment for anxiety-related disorders, it is minimally effective as a standalone treatment for drug-related disorders. This led to our research program to build a translational animal model of cue exposure therapy for the discovery of novel strategies that could enhance the effectiveness of extinction training to inhibit cocaine relapse. We begin our experiments by exposing animals to a long baseline history of cocaine self-administration, followed by extinction training, whereby presentation of drug-paired cues is contingent on lever pressing. This approach is different from most preclinical studies that only extinguish lever pressing. That is, in other studies, both drug and drug-paired cues are omitted during extinction training. This latter approach is not a model of Q exposure therapy. In our experiments, reacquisition of cocaine self-administration is evaluated to model cocaine relapse. Again, this is different from most preclinical studies that study relapse via Q or prime-induced reinstatement of drug-seeking levopressors. These latter approaches used by others are not models of relapse to drug use. A critical feature of our work is the use of a second-order schedule of Q presentation and drug delivery during the fixed interval component, which is a drug-free interval. Every 5 responses results in a 2-second presentation of the drug paired Q. After the fixed interval ends, the completion of 5 responses results in a 2-second presentation of the drug paired Q plus drug delivery. In this way, each session begins with a Q-only component, serving as a retrieval Q period for cocaine memory. Extinction sessions are the same as drug sessions, except that saline is delivered instead of cocaine. For the first extinction session, cocaine memory is retrieved during the first drug-free interval, but the anticipated injection of cocaine is withheld and is followed by extinction training. Now you might ask, why do we do all this? It has been shown that exposure therapy for relapse prevention works best if drug retrieval cues are incorporated just prior to extinction training. Shui and colleagues found that a brief period of heroin memory retrieval prior to Q exposure therapy reduced heroin relapse and craving up to 184 days later. Other important features of our translational model include 1-hour extinction training sessions, which is the typical length of a Q exposure therapy session in people. Extinction sessions that are spaced at weekly intervals. This is the optimal spacing for Q exposure therapy, which is not as effective if spaced too closely together or too far apart. And lastly, use of pharmacological and behavioral cognitive enhancement strategies during extinction training. A pharmacological target for cognitive enhancement in extinction learning is the NMDA receptor. Drugs such as D-cycloserine, which is a glycine site partial agonist, and the Rho and ORG compounds, which are inhibitors of glycine transporter 1, enhance NMDA-mediated glutamate transmission to improve cognition. For our behavioral strategy, we use environmental enrichment as a brief intervention and not as a housing condition. These are 4-hour experiences that we refer to as RATCAP. I have a video here, which I can show you how they interact. As you can see, EE provides the opportunity for physical exercise, that is, there's running wheels in the enclosure, as well as cognitive stimulation through the use of toys and tunnels and foraging for Fruit Loops, which are sprinkled and hidden throughout. And then, of course, social interaction, where rats bring 3 or 4 together. These rats normally live in isolated housing conditions. Nothing as nice as RATCAP. So the conceptual framework for using glycine-based drugs and an EE strategy relates to the fact that extinction training itself and learning increases AMPA-GLUA1 surfix expression, which induces synaptic plasticity that is needed for learning and memory. LIT1 inhibitors increase glycine binding at the NR1 subunit of the NMDA receptor to enhance AMPA-GLUA1 surfix expression as well. And environmental enrichment increases BDNF and TrkB receptor expression, as well as NR2B subunit expression to enhance AMPA-GLUA1 surface expression as well. So these strategies alone and combined might facilitate extinction and inhibit cocaine relapse via the primary cellular means by which synaptic plasticity and learning and memory unfold. The next series of slides show some key behavioral findings examining the effects of ORG alone, EE alone, and EE plus ORG combined with extinction training. The ORG experiment involved 4 different treatment conditions. Rats and monkeys were first trained to self-administer cocaine at a moderate dose. That was 0.3 milligrams per kilogram in rats, 0.18 milligrams per kilogram in monkeys. And they did this for a minimum of 7 weeks. And then they were given 3 weekly extinction training sessions. During extinction, either vehicle or ORG was administered prior to the extinction training session. In addition, 2 types of control conditions were used to examine the effects of ORG in the absence of extinction training. The first was something we called weekly no extinction sessions. The animal was put into the operant chamber, but the levers were retracted and no stimulus lights or saline infusions were given, or weekly cocaine self-administration sessions, which were the same as baseline sessions. And then after extinction training, there were 15 daily cocaine reacquisition sessions, starting 1 week after the last extinction for control session. This slide shows that ORG did not modify the rate of cocaine Q extinction learning in either rats or monkeys. In the rats, animals treated with ORG3 or ORG 7.5, their rate of extinction learning was the same as animals that were treated with vehicle. And the same thing was true of monkeys who were treated with ORG 1 milligram per kilogram. However, ORG did inhibit reacquisition of cocaine self-administration. Rats that received vehicle treatment, that's the white circles here, during extinction, they relapsed immediately to baseline rates of cocaine self-administration. When they were treated with ORG3, that's the gray circles here, they had reduced reacquisition through session 12, and then they were treated with a slightly higher dose, ORG 7.5, that's the black circles. They had reduced reacquisition through session 9. This is not surprising because there is an inverted U-shaped relationship between cognitive enhancement and dose of ORG 24598. In monkeys, the results were similar. The monkeys treated with ORG1, the black circles here, they reduced reacquisition to the same degree and length of time as ORG3 in rats. If they got treatment with vehicle during extinction, they relapsed immediately to baseline rates of cocaine self-administration. Kind of consistent with the clinical literature showing Q exposure therapy or extinction training alone is not effective in reducing relapse behavior. These are the results from those control conditions. When ORG is administered prior to either the weekly no extinction sessions or the weekly self-administration sessions, there was no effect of ORG on reacquisition in rats or monkeys. Here, rats, animals returned to baseline rates of cocaine self-administration within 1 to 2 sessions, whether it was a no extinction training or which we call abstinence or self-administration weekly sessions. And the same thing in the monkeys. These monkeys relapsed if they didn't have extinction training with the ORG. So what this suggests is that extinction training is needed in order for ORG to inhibit cocaine relapse behavior. This is the EE experiment, and this also involved 4 different treatment conditions. Rats once again, were trained to self-administer a moderate dose of cocaine for 7 weeks, and then given 3 weekly extinction sessions. EE was provided in 4-hour periods, both 24 hours before and immediately after each weekly extinction session. These rats were compared to rats that received extinction training but without EE. That is, they were put back into their individual home cages after each extinction training session. Control conditions were used to examine the effects of EE in the absence of extinction training. That would be this group here. So they had an abstinence session or no extinction session to examine the effects of just EE. And these were compared to rats that had these no extinction sessions but did not have EE. And then there were 15 daily cocaine reacquisition sessions that started 1 week after the last extinction session. This slide shows that EE facilitated the rate of cocaine Q extinction learning. On the left here, rats that received EE with extinction training, that's the white circles, they reduced their responding sooner and to a greater degree than rats that just received extinction training alone, which is the black circles. EE also reduced reacquisition of cocaine self-administration. Rats that just received extinction training, the black circles here, they relapsed immediately to baseline levels. But if they received EE with the extinction training, the white circles here, reacquisition was reduced through session 13. Thus, the effects of EE on cocaine relapse are just as robust as ORG3 in the male rats. Under control conditions, when EE was administered prior to weekly no extinction sessions, there is no effect of EE on reacquisition of cocaine self-administration. These animals relapsed immediately to baseline levels. So just like with ORG, these findings indicate that EE also has to be provided with extinction training to inhibit cocaine relapse. Now, a critical question is whether EE can prevent what's called the context dependency of extinction memory retrieval. Extinction training is known to work well for reducing fear if conditioning, extinction, and retrieval all take place in the same context, a so-called AAA design. However, if extinction training takes place in a novel context that is different from that used for conditioning and retrieval, then extinction training does not work well for reducing fear. This is what's called an ABA design. This context dependency for relapse prevention also applies to cocaine Q extinction training in context A versus context B. Past research has shown that pairing extinction training with cognitive enhancing pharmacological agents can overcome this context dependency for relapse prevention. This has been shown with D-cycloserine, L-DOPA, as well as HDAC inhibitors. Therefore, this experiment was conducted to determine if EE could prevent the context dependency of extinction memory retrieval during relapse testing. And this is important to determine because treatment-seeking cocaine users would receive Q exposure therapy in a clinic setting, which is a context B, rather than in the original drug-taking context, which is a context A, where they would be more prone to relapse. So 3 groups of rats were trained to self-administer a moderate dose of cocaine in context A for 7 weeks, and then given 3 weekly extinction sessions, either in context A without the EE protocol, in context A with the EE protocol, or in context B with the EE protocol. Then there were 15 daily cocaine reacquisition sessions, all in context A, starting 1 week after the last extinction session. This slide shows that EE facilitated the rate of cocaine Q extinction learning, whether extinction learning took place in context A, which is the white squares here, or in context B, which are the gray squares, compared to the no EE control. However, EE reduced cocaine relapse only after extinction training in context A. Rats that received EE plus extinction in context A had reduced reacquisition through session 15. And rats that received EE and extinction in context B, these gray squares, they relapsed immediately to baseline levels of cocaine self-administration, as did the no EE control group. Thus, EE interventions during extinction training were not able to prevent the context dependency of extinction memory retrieval or relapse prevention. The next series of slides shows the EE plus ORG experiment and finally addressing the question, are there sex differences? Rats now were trained to administer a high dose of cocaine, 1 milligram per kilogram, for 7 weeks, and then given 6 weekly extinction sessions because of the higher state of cocaine dependence. That is, it takes them longer to extinguish. And then rats either received extinction training without any added treatments, extinction training with EE plus ORG, extinction training with EE alone, and extinction training with ORG alone. As before, extinction training or EE was provided in 4-hour periods, 24 hours before and immediately after each weekly extinction session. Then there were 15 daily reacquisition sessions starting 1 week after the last extinction session. Although not shown, it is important to point out that during baseline, lever responses and cocaine intakes were not different between groups or between sexes prior to the extinction treatments. During extinction in male rats on the left here, EE plus ORG3, the black diamonds, facilitated the rate of extinction learning compared to control, which are the white triangles. EE alone, which is the gray squares, and ORG3 alone, the white circles, did not facilitate the rate of extinction learning compared to control. In the females on the right here, ORG3 alone did not facilitate the rate of extinction learning, but ORG7, the hexagons, did, suggesting that ORG 7.5 is a behaviorally active dose of ORG in females that's comparable to ORG3 in males. Thus, there were sex differences during extinction. In a direct comparison between sexes on the left, when there was no added treatments, females, the black circles, extinguished faster than males, which are the white circles. And on the right, with EE plus the behaviorally active dose of ORG, females, the black circles, also extinguish faster than males, which are white circles. Despite this benefit in the females in terms of facilitation of extinction learning, the EE plus ORG combination reduced cocaine relapse in males, but not female rats. In the male rats on the left, EE plus ORG3, the black diamonds, reduced reacquisition across the 15 sessions compared to control, which are the white triangles. EE alone, the gray squares, and ORG3 alone, white circles, did not reduce reacquisition. These groups relapsed within 1 to 3 reacquisition sessions. In female rats on the right, there were no group differences during reacquisition, with neither EE plus ORG 7.5, that's the hexagons here, nor EE plus ORG3, which are the black diamonds, reducing reacquisition of cocaine self-administration. These two EE plus ORG treated female groups returned to baseline levels of cocaine self-administration immediately. There was a slight delay in the return to baseline in females that did not receive any added treatment. That was these gray squares or gray triangles here. It's important to note that there's large variance in the EE plus ORG 7.5 group of females. This was due to one rat that was at 100% of baseline during the first 3 reacquisition sessions, but then afterwards increased its responding from 200% to 1200% above baseline thereafter. We checked the function of the rat's catheter, and it was very functional throughout, so we kept that rat's data in the analysis. But even if we remove this one rat from the analysis, no between group differences remained, suggesting that this procedure did not work in females where it worked so well in the males. So thus, there were sex differences during reacquisition as well. In a direct comparison, on the left here with no added treatment, females made fewer relapse responses, the black circles, and had lower cocaine intake, the black bar, than males, which is the white circles and the white bar. This suggests that extinction training alone has a small benefit in the females. With EE plus the behaviorally active dose of ORG on the right here, females made more relapse responses, the black circles, and had greater cocaine intake, the black bar, than the males, the white circles and white bar. And this suggests that EE plus ORG is ineffective in females and may actually counteract the small benefit of extinction training alone. So to summarize the pharmacological and behavioral determinants, we showed that facilitating the rate of extinction learning is not necessary for relapse prevention. The important factor is likely facilitation of extinction memory retrieval. ORG alone or EE alone inhibits relapse for a moderate dose of cocaine, that is 0.3 milligrams per kilogram in rat. EE plus ORG is needed to inhibit relapse for a high dose of cocaine, that is at 1 milligram per kilogram in rat. EE does not prevent the context dependency of extinction memory retrieval. And a question is, can EE plus a GlyT1 inhibitor prevent context dependency? And this study is currently in progress. EE and ORG are effective for inhibiting relapse only when combined with extinction training. And we showed that cocaine relapse is immediate if no extinction training is used. Lastly, this strategy works best and well in males, but not female rats, despite evidence that ORG 7.5 is behaviorally active in females. An important question is, why is this strategy ineffective in females? We are hoping to answer this question as we build our molecular model in animals. And the next series of slides shows some key molecular findings regarding neurosubstrates of extinction learning, as well as changes in GLUA1 and TREC-B expression related to cocaine self-administration, extinction training, and extinction training with EE that may contribute to relapse and relapse prevention. CFOS expression was used to determine which brain sites are activated after extinction learning. CFOS expression in the prelimbic prefrontal cortex and the basolateral amygdala are selectively activated by extinction learning, cocaine Q extinction learning. If they had saline extinction or had cocaine but no extinction, the levels of CFOS were not expressed as highly. Exposure to cocaine cues and saline cues, that is the cocaine extinction and the saline extinction, enhanced CFOS expression in the infralimbic prefrontal cortex and dorsal hippocampus, suggesting these sites are important for processing Q salience during extinction training. And lastly, lever pressing during extinction training correlated with CFOS expression in the nucleus accumbens core, suggesting the site may mediate motor behavior during extinction training. Knowing that these are neurosubstrates of extinction, when we started looking at measurements of GluA1 and TrkB, we focused on changes in the basolateral amygdala, the dorsal hippocampus, the ventral medial prefrontal cortex, and the nucleus accumbens. First, we discovered that chronic cocaine self-administration reduced GluA1 and phosphorylated GluA1 expression in the basolateral amygdala, and also reduced GluA1 expression in the nucleus accumbens without changes in the dorsal hippocampus or the ventral medial prefrontal cortex. And that was comparing rats with a cocaine history only to rats with a saline history only. In these same brain sites, cocaine Q extinction training with or without EE, that's on these two right side in each graph, increased GluA1 in and phosphorylated GluA1 in the basolateral amygdala, also increased GluA1 in the nucleus accumbens, the dorsal hippocampus, and the ventral medial prefrontal cortex, compared to groups that had no extinction training with or without EE. That's the first two groups in each of these graphs. Besides increases in GluA1, EE in combination with extinction training in context A also caused an increase in TrkB expression in the dorsal hippocampus and increased TrkB signaling in the ventral medial prefrontal cortex as well as the nucleus accumbens. And then lastly, there is a very large behavioral and molecular study in progress in male rats to determine what mechanisms may underlie the beneficial effects of EE plus ORG with extinction for cocaine relapse prevention. This group involves 9 different groups of rats and 7 protein targets. Tissue analysis is ongoing. So some rats were euthanized after extinction training, and other rats after extinction training went on to have reacquisition of cocaine self-administration daily for 15 sessions. Here's a sneak peek at the behavioral results so far. On the left, during the 6 weekly extinction training sessions, the extinction learning was the fastest in the EE plus ORG, the black triangles, that is, it was reduced soonest compared to EE alone versus extinction training alone. On the right is reacquisition of cocaine self-administration, and when averaged over the 15 sessions, the cocaine self-administration was significantly reduced by EE plus ORG compared to no extinction training control. If they had extinction training with EE or just extinction training, these groups were no different than the no extinction control. So these data speak to the reproducibility of our earlier findings in male rats self-administering a high dose of cocaine and the need for EE plus ORG to reduce cocaine relapse when they are self-administering this high dose. As we analyzed the brains of these male rats, along with the brains of the female rats that received EE plus ORG 7.5 with extinction training, we may gain insights into why this strategy did not work in the females. Here are some ideas from past research on why females might be different from males. First, EE does not enhance BDNF expression in the hippocampus and frontal cortex of female mice as it does in males. We had shown, for example, that the only animals that had reduced reacquisition of cocaine self-administration with EE were the male rats that got extinction training in context A, and context A was the only condition that increased TrkB, the receptor for BDNF, in the dorsal hippocampus. Second, genetic deletion of GlyT1, which can be thought of as a proxy for glycine transporter 1 inhibition, causes a resistance to extinction in the female mice, but enhances extinction in males. And this is similar to what we saw, that the females that got extinction training alone, there's a small but slight reduction in relapse behavior, at least temporarily. That's something that's never seen in the males. And when they got the E plus ORG, that actually seemed to prevent this small benefit of extinction training, suggesting sort of a more of a resistance to extinction, having had the ORG with the EE. And then the third possibility is the mnemonic properties of GlyT1 inhibitors in AE may be influenced in a sex-dependent manner because females have higher basal levels of NR1 and NR2B subunits than male rats. This could act counterproductive to adding something like EE and GlyT1 inhibitors that act on these same subunits. So, like I said, when we get that huge study completely analyzed, we'll get to see which of these factors may be important. So we predict that the benefits of EE and ORG involved enhanced GLUA1 and TrkB signaling in these same brain sites in male rats, and that these changes will not occur. To summarize the molecular determinants, we showed that chronic cocaine self-administration disrupts GLUA1 signaling in basolateral amygdala and nucleus accumbens. Extinction training plus EE helps to reduce cocaine relapse by enhancing GLUA1 signaling in the BLA and nucleus accumbens, as well as by enhancing TrkB expression and signaling in the dorsal hippocampus, ventral medial PSC, and nucleus accumbens. We therefore predict that the benefits of EE plus ORG involved enhanced GLUA1 and TrkB signaling in these same sites in male rats, and that these changes will not occur in female rats. So collectively, this body of work points to two translational challenges on the horizon. The first relates to the ineffectiveness of these strategies in females. And second is the possibility that the EE plus ORG combination also will not prevent the context dependency of extinction memory retrieval, which is a problem we're using this in a clinic setting. To address the first challenge with females, we are making lentiviruses that overexpress or silence GLUA1 or TrkB, which are two protein targets we demonstrated to be important for the effects of extinction training and EE for cocaine relapse prevention in the male rats. In vivo validation studies, lentivirus GLUA1 GFP overexpressed GLUA1 in the basolateral amygdala by 200% 7 weeks after infusion. This is the time frame between the start of extinction training and the start of reacquisition testing. The lentivirus TrkB GFP overexpressed TrkB in the dorsal hippocampus by 150% in the same time frame. So these two lentiviruses are ready to go. In vitro validation studies, lentivirus short hairpin GLUA1 decreased GLUA1 expression in cultured neurons by 50%. We are currently making a lentivirus short hairpin TrkB, and then both will be validated in vivo. The thought that we can get over 100% increased expression and silence 50% expression will make these good in vivo tools. The questions we want to ask are, will silencing GluA1 in the basolateral amygdala or TrkB in the dorsal hippocampus prevent EE plus ORG from inhibiting cocaine relapse in male rats? Second, will overexpressing GluA1 in the basolateral amygdala or TrkB in the dorsal hippocampus mimic the effects of EE plus ORG for inhibiting cocaine relapse in the male rats? And then lastly, will an overexpression virus strategy benefit female rats who do not respond to EE plus ORG? If so, then an alternative way to induce these kinds of plasticity changes during Q exposure therapy in females may be necessary. Regarding the second challenge on the horizon, if we find that EE plus ORG does not prevent the context dependency of extinction memory retrieval, this raises the question of whether virtual reality can be used to recreate drug-taking context in a clinic setting while patients undergo Q exposure therapy, thereby turning an ABA situation into an AAA situation and assisting in relapse prevention. And I just want to thank you for your attention, and I'm happy to answer any questions you may have.
Well, thank you very much, Kathleen. That was a very interesting series of studies, and so we can open it up to questions. We have some time here. I just wanted to start off in terms of the sex differences seem to be pretty pronounced, and
Yes.
I wonder in terms of the cyclicity of the females. I don't know if you've done any parametric studies of that over the course of their training and on their 4-day cycle, you know, they'd be in different states in terms of possibly getting kind of more or less reinforcement. So might they be resistant to the effects of the EE and the ORG effects because they've had kind of this partial reinforcement extinction effect?
Yeah, I didn't monitor the estrous cycle in these females, but in a past study that I had done, I think in 2007, compared adult cocaine self-administration in adult males and adult females. And when they were trained to self-administer this high dose, 1 milligram per kilogram, looked at cocaine dose response functions in the males versus females, and they were not different. That is that the amount of cocaine that they self-administered at each dose was the same. So there were no sex differences. So with a high dose, you tend not to get sex differences in intake, even though there is, and it didn't depend, and there were no differences at each phase of the estrous cycle as well, because we did monitor that. So I think, you know, the fact that the estrous cycle did not influence cocaine self-administration when a high dose of cocaine was being self-administered, I didn't worry about the estrous cycle having some impact. And these females were very stable day to day to day to day, because they have a long 7-week baseline history.
Oh, right.
Lots and lots of days to see them self-administer. And it's not like we saw a sort of a cyclic effect. It stayed very stable.
Okay. Well, thank you. Jed, do you have a question?
Yeah. Thanks. That was a wonderful talk, and integrating the neurochemical mechanisms with the behavioral findings is really very, very interesting. And I wonder, on that note, that important theme about the context dependency of extinction, right? It's a good idea that one that you suggested, maybe VR could be used to bring the external environment into the lab. And I wonder, though, have you thought about, or has anybody done the opposite of trying to bring the lab to the real world, in the sense that
that would be the ideal.
Yeah,
well, it's not going to happen.
Well, I'm thinking, is it possible to transfer the control of the extinction process to, let's say, an internal cue, a taste cue or interoceptive cue, or some drug discrimination cue that some other drug would produce. So in other words, they're in a different interoceptive internal state or sensory state when extinction's happening, and then they can carry that, whatever is giving them that, a taste of a distinctive food or drink, distinctive
Carry the cue back. But I think, but what you have to though is extinguish the drug paired cues. So you're talking about transferring the drug paired cue to a different type of cue that can. Well, but they're going to go back, and the drug paired cues are going to be in their home environment.
Well, yeah. So let's suppose that, you know, by infusing drinking water with a certain taste, that makes the context different during extinction. So then, when they're extinguishing, when the cocaine cues are being extinguished, it's in the presence of, oh, now I'm in a situation where water tastes different than it always has. And then, when you go in the real world, though, you make your source of water that same water flavor that was there during extinction. So now the real world feels like the lab context because you've now made a new contextual cue that you can carry around with you.
Okay. Oh, I like that. I like that idea. So yeah, you can have to artificially create one, and that, yeah. Oh, I like that idea a lot.
Well,
Great. Other questions, people? Okay, I have another question. So in terms of this, you know, concentrating on the EE benefit, so I've always been fascinated in terms of the relationship of HPA axis activation and relapse and maintenance of drug abuse. And so is there a possibility of a kind of a conditioning of HPA arousal with the act of taking a drug? And then the fact is that whenever you put an animal in a enriched environment, they actually have a lot of different things they might relate to HPA arousal, and kind of broaden out their learning of, well, now I'm feeling aroused, and it's related to this toy or this other animal or some other environmental condition, rather than having it so narrowly focused on hit the lever, got the drug, and then that's related. So you kind of extinguish the HPA arousal from the drug taking experience. And then that might actually be related to your observed sex differences with at least the EE and the extinction, because whenever you have the conspecifics, I assume it's all isosexual. You have all females with females.
Right.
Females. And so maybe the males find that a little bit more HPA arousing in terms of battling it out with the other males, whereas the females maybe less so. I don't know much about
well,
the social nature on that.
I was surprised that the EE, well, we only looked at EE plus ORG in the females so far, that it didn't work, because when they were in EE, the females seemed to get much more out of the EE than the males did, where they would immediately go in and just start interacting with each other. And before we were doing these 4-hour periods, we would once a week do these brief 15-minute periods just to make sure that we could get compatible rats together. Because once in a while with the males, there'll be an extreme bully that you don't want to put in there. And so sort of weed that out ahead of time.
You call that the Trump rat.
I like that. And so, but the female, the males would go in there and they'd sort of like go to their own corners and over time start interacting. But, the females definitely interacted so much more. So, in some respects, the EE may have been more arousing for the males than for the females, because there was a bit of stress in there. And of course, arousal and a little bit of stress is needed for effective memory consolidation.
Right.
And so that may be why that worked so well in the males, because the EE was a bit more stressful for them than it was the females. And the females was more sort of like even though they lived isolated, when they got together, as if they were never isolated. during that time. It was sort of like business as usual. Oh, other females, let's go play, let's go do this. Let's share this environment. So, so maybe, one way to sort of make, exposure therapy with cognitive enhancing manipulations better in females would be to add a little bit of stress to the situation, a little bit of arousal.
Right. That's divorced from the actual drug taking experience. Yeah. So, other comments or questions? I actually have more, but I don't want to dominate. So you found that the EE and the ORG basically had kind of additive effects. I mean, can you differentiate? Does the ORG enhance the effect of the EE, or are those two kind of mechanistically different and they just
they're mechanistically different because what they're targeting and so you're coming at AMPA trafficking and surface construction, and there's many different ways to enhance that AMPA GLUA1 trafficking to the surface, and they're coming at it by different ways, which it's not like they're working on the same target, in which case you might not expect them to work in a facilitative way, working better together than each one individually. Because if the GlyT1 inhibitor is doing this coming through the NR1 subunit, the glycine site on the NMDA receptor, and environmental enrichment is coming through the BDNF track B side to get to enhance, surface expression of GLUA1. It's like two opportunities to do the same thing. And so it adds together.
Okay.
Is what I'm thinking.
Good.
We'll see that when we get all that tissue analyzed, because we have all the controls. There's the extinction and abstinence with just EE, the extinction and abstinence with just ORG, the extinction and abstinence with the EE plus ORG, and then there's cocaine alone, then yolk saline. Yeah. So we have, took 9 groups to control for everything, but it's going to be a very important study when it's all done to give us mechanistic insights.
Yeah, Jed, you had another question?
Yeah, just in terms of, if you can work out, overcome the obstacles of sex difference and context dependency and all that, how far would these drugs be from the clinic in terms of tox testing and so forth?
Well, for several years, GlyT1 inhibitors were being developed by several pharmaceutical companies as adjuncts to give in people with schizophrenia, adjuncts to their antipsychotic medication to improve cognition, part of that sort of whole matrix design that they were doing at one time. And most of these companies sort of abandoned that because and that seemed to, at the time, to be the only application of these GlyT1 inhibitors, because when they went through clinical trials, they didn't show that these drugs offered any improvement for cognitive enhancement over just having their antipsychotic treatment, typically a atypical antipsychotic drug. So they sort of abandoned that. But that doesn't mean, so they're only being evaluated in people with a disease, a serious disease, schizophrenia. But this doesn't minimize the fact that they do have cognitive enhancing effects, and they could be used, something like this. I'm not saying it's not an everyday treatment sort of medication, but the medication is being used very strategically with these weekly extinction training sessions to enhance that extinction memory retrieval. But I think a lot of pharma companies, because they were developing it as an adjunct for schizophrenia cognitive enhancement, and that didn't work, and they abandoned it. So these drugs are just sitting there.
Well, yeah, but they may have a lot of tox, at least preclinical tox data, and some human.
But several of them have that. Like Kaufman LaRoche has gone through clinical trials, up to phase 3 clinical trials, for example, with their GlyT1 inhibitor. So to get to phase 3, it had to pass all the tox stuff.
Definitely had to pass that. And in terms of like seizure risk, is this not like this
very low? It's,
yeah.
These newer GlyT1, sarcosine was the original GlyT1 inhibitor, and that was not forebrain selective, and that had some respiratory issues when it would hit glycine site in the brainstem. But these newer ones are forebrain selective, and they're only hitting the glycine site in forebrain structures and cortical structures, and so they tend to be side effect free. And if you're using these for cognitive enhancement, low doses are actually better than higher doses, because there's this inverted relationship between glycine levels, synaptic levels of glycine, and cognitive enhancement. Too little or too much is disruptive to memory consolidation and optimal, and this is achieved with low doses, which would sort of reduce the toxicity.
Well, that sounds very promising. So do you know of any other data that would suggest that there would be more general effect across drugs of abuse, say with opiates or nicotine or alcohol, in terms of this approach?
Right. No one else has really used this approach preclinically, but in a sense, there's some clinical data out there using Q exposure therapy, for nicotine has been looked at, and cocaine has been looked at, alcohol and alcohol cues have been looked at, and there's some work with D-cycloserine, and a lot of that clinical work is negative in terms of the findings, but there's I consider a lot of problems with the way they did those studies. And if they don't follow sort of like the best practice for using Q exposure therapy, like they were lumping, extinction training sessions every day for 3 or 4 days, and giving, rather than spacing them out, not having retrieval cues for the drug memory prior to, and then only having extinction after they've been hyper aroused by the drug cues. And then only D-cycloserine has been looked at. Even in our own work in animals, we did get benefits of D-cycloserine in the rats and monkeys, but the GlyT1 inhibitors work so much better. A much longer relapse prevention with GlyT1 inhibitors than DCF.
Yeah, I mean, I see that in a lot of ways in the early human studies that are underpowered and not optimally done, and then they come up negative, and that puts a kibosh on the whole endeavor, which is
no, it has to do with I have not been able to find anything about sex differences in Q exposure therapy, whether for drug or for anxiety disorders, because these studies, even though they include females, are tremendously underpowered, and no one has looked for sex differences, which sort of surprised me. So I don't know what the clinical situation is regarding Q exposure and potential sex differences for that.
Well, if anyone out there has some, please, I'll share it with the group. So,
okay.
I want to thank you again, Kathleen, for.
Okay, well, thank you for inviting me. It was great talking to all of you.
Very nice discussion we've had. So I want to welcome everybody back on April 14th, where Nicole Shram-Cepeda will be talking to the group. And yeah, keep well till then. Thanks.
Okay, take care. Bye-bye.